Well, welcome everybody to the 6th annual CEMB Summer REU Research Symposium. This is the culmination of a major project from a major center. As you all know, CEMB is the American investment, the National Science Foundation's major investment in the science of mechanobiology. And this is a program that's joint between Washington University and the University of Pennsylvania with several satellite sites. We're very pleased to have people from many of the satellite sites online today. This is also a program that's been run in collaboration with industry. In particular, we're very fortunate to have a strong relationship led by Phil Taylor with the Bayer organization here in town and worldwide. So the program has been put together by a number of talented individuals. We have in the room here. The room is here to capacity. Please be sure you spread out a little bit. Don't violate our COVID-19 regulations. But we're overflowing in the room here. But amongst these people in the room are the bench mentors and the faculty who have helped put this together to help get these outstanding students where they are. We also have in the room the people who have really put this together. So before we begin, I want to acknowledge the important role that this program has involved. You'll hear about this program a little bit more from Patricia Widder, the site coordinator for it, in just a moment. She's been working in concert with bench mentors, with Ryan Calcott, Ryan Richardson, Jose Almeida, Dr. Ethan Hoppy, Natasha Bilkey, and Morgan Lee, and Shanjun Peng to put this together. And you'll hear about the research they've done with these bench mentors, with the faculty of CEMB, and also hear a little bit about some of the guidance and interactions that these students have been very fortunate to have with true world experts in their respective areas from the Bayer Corporation. So actually, we're very pleased that Lauren Klein, Christopher Van Horn, Jung Kim, Monica Van Gordon, Ross Bickelmeyer is here, and Manasi Petha, all online from the Bayer here in town and around the world. I see Christopher Van Horn's actually joining from Germany. We have Giska Giska Ramadita joining from Indonesia. Truly a worldwide event. So to begin the event today, I'm very pleased to introduce someone who's a stranger to none. This is Dr. Professor no less, Patricia Widder from Washington University, who's been the site coordinator for this event. Please welcome Patricia Widder.
Thanks so much, Guy, and welcome to everyone who's here with us today. I'm so pleased with the work that has been done by these 6 students this summer, and we are very thankful for the contributions of their bench mentors and their PIs and the others who have helped them. We've had a great summer out within the labs and outside the labs. The students have participated in extra workshops and educational opportunities. We've talked about making scientific presentations, how to create a good poster, how to submit an abstract. We've talked about networking and resumes and mentorship. And we've done some fun things too. The students went to the St. Louis Arch and I think probably a couple of other places that we don't know. We also were one of the first groups to return to touring in person at the Danforth Plant Science Center here in St. Louis. And that was a really great experience for us to see what St. Louis has to offer in the field of plant technology. We are so thankful for the Bayer mentors who were unfortunately unable to meet with us in person, but I know that they met with our students regularly virtually throughout the summer. So we're very grateful for that. Before we get to the presentations, I'd like to introduce, I think it'll be Megan O'Donnell, who has a few words for the students about some of the housekeeping at the end of the summer here. So go ahead, Megan, if you're ready.
Hi, everyone. My name is Megan O'Donnell, and I'm with the College Research Evaluations Team, or CREST, and I'm with Arizona State University, and we are the new external evaluator. So I just wanted you to be able to put a face with the name that you'll be seeing, because we're going to send you surveys and try to schedule some focus groups to get your insights on how this program has impacted your skill levels and, you know, any feedback you can have on how we can improve the program so that it's even better next year. So again, my name is Megan. And there will also be 2 other people who might be contacting you that are part of my team, and that's Karen Gordon or Wendy Barnard. So you might see their names. And yeah, so we will be in touch soon with the surveys and to schedule some focus groups for next week. So thank you very much. And we're really excited to be part of the team. So thanks.
Thank you, Megan. We should appreciate the work that the CEMB does to support all of the efforts. All right, let's end then with our presentations. We're going to, let me get a poster up. We have Julia Behlmann is our first presenter. She's from with the University of Tulsa and has been in the Haswell lab. And you can say that again, Julia, if you want to, because it's probably part of your beginning.
Thank you, Patricia. Can you guys hear me? Okay, so hi everyone. My name is Julia Behlmann. I am a rising junior at the University of Tulsa, and I am a mechanical engineering student. Through the mechanobiology program, I was fortunate enough to be in Dr. Elizabeth Haswell's plant biology lab. And her lab asked the question of, how do plants perceive forces? It's like, well, how do humans perceive forces? We have touch that sends signals through our nervous system. Our ears pop when the pressure changes. And so in plants, they don't have the nervous system. So sometimes they respond to touch, but also one of the ways they perceive forces is through mechanosensitive channels. Now, mechanosensitive means that there's mechanical stimuli, again, can be touch or even osmotic pressure, and this creates tension at the cell membrane. So that membrane will then pull open a pore in a channel. So we are interested in the mechanosensitive ion channel, OSCA2A. Because it is an ion channel, OSCA is significant because it is specific to calcium ions. And we know calcium has been implicated in drought response. And so we were like, okay, let's look at OSCAs and then in plants, because we know in humans, they deal with like swelling and other similar responses. So we said, okay, let's look at OSCAs in plants. Well, let's use moss, because moss is incredible in the fact that it can dry out and then rehydrate itself. And that's something that's incredibly unique to moss, because other model plants like Arabidopsis or rice are not nearly as resilient when it comes to this. So if you can see in figure 1, this mouse not going to work. Okay. In figure 1, we have a visual representation of an OSCA channel. It is at the membrane, and you see calcium ions can flow across it when the channel is open. This means that there's already been tension at the membrane and the pore is open. If you look at figure 2, we see an evolutionary tree. Now, the ones in light blue are the 15 moss homologs that are the OSCAs that we were interested in across 3 different clades. Clades just mean that they share a common ancestor. And so the reason we're looking into OSCAs in general is because they're widely apparent across species. And so we said, okay, let's remove one of these genes and see what happens. Well, in clade 2, there's only 2 OSCAs. So removing 2A allows us to remove 50% of the OSCAs in that clade. And so we would assume to see a phenotype. So one of the goals of our research was, okay, let's remove OSCA2A and see how it's different than the wild type moss that occurs in nature. Another goal we had was, if you look at figure 2 again, does OSCA 1A, B, and C? We said, let's eliminate 3 OSCAs out of the 6 in that clade, because again, that's 50%, and we'd hope to see a phenotype if we knock out approximately 50% is the goal. So for the materials and methods, if you see in figure 3, we used CRISPR target sites to knock out the genes that would create the mutant lines for A, B, and C. So the 2 triangles, the 2 orange triangles, are the CRISPR sites that we identified, and then we used gateway cloning to knock out the majority of the pore. Even though not the entire pore will be removed, because the majority is knocked out, it'll be disrupted and it will not function as a pore because the channel can no longer open. For phenotyping the clade 2 mutants, we used different salt plates such as mannitol, NaCl, KCl, and we also tested on gravity plates, which are square plates, and you set them vertically and they grow towards the light. And then we also tested on water agar. And the reason you test on different salts is because you want to see, okay, if we're giving the moss samples too much salts and it can't have access across this one pore, what kind of stress will that occur? What will survival look like? And then with the water agar, the reason we tested that one is it's the opposite, where there's not enough nutrients, there's not enough salts. And we wanted to see if the wild type would survive more than the mutant lines. The mutant lines we have from 2A, we are called Delta Osca 2A-45 and Delta Osca 2A-50. What that means is that they are genetically modified and they are missing the majority of the pore from 2A. So if you look back at figure 2, the 2A that is highlighted, and then if you look at figure 4, the two middle panels are missing that 2A gene. So back to figure 4, this is the desiccation assay, which is the last part of the methods. So we use a desiccation assay where we put different amounts of ABA, which is a plant growth hormone, and we let the samples incubate on that overnight and then dry out the next overnight on completely sterile plates. Then they got all thin and green and crispy. And then we let them rehydrate on regular BCDAT media for a week and recover. So if you look at figure 4 again, in the panels in part A, the black outline was all green after it dried out. It was all green and crispy. And then when we put them to rehydrate, only the area that is green currently is still alive and the rest is white, which means it's dead. And what's incredible is that the wild type, we used Fiji, determined the percentage of survival with thresholding. The wild type was 19.8% survival compared to 11.3% in Delta Osca 2A-45 and 8.99% in -50. So this shows that Osca 2A channel must be important in drought response or drought survival, because it was over double survival in the wild type than the mutant lines, or at least it suggests this. We also noticed that the wild type appeared less dense at the edges under the microscope than Delta Osca 2A-45 and -50. If you come up to figure 5, you'll see that on the left panel for the wild type, it's a bit more loose at the ends, a bit more spread out. 50, you can see pretty clearly that the edges are much more packed together, and we noticed 45 is more similar to 50 than the wild type. We also said that the wild type appears greener on water agar compared to Delta Osca 2-45 and -50. That's in figure 6. So if you see on figure 6, again, the left panel is the wild type. These are brighter green. Even though moss needs more nutrients than the water agar provided, the wild type was still able to survive better than the Delta Osca 2A-45 and -50, as you can tell by the darker brown middles in both 45 and 50. There was no noticeable difference on the other assays mentioned, and that's important to test because we wanted to see too much salts didn't provide that much of a difference, but not enough salt in the water agar definitely did. And then we did make progress on making a 2-fragment recombination for Delta Osca 1C. This is the gateway cloning process we did because we've just started generating the mutants from clade 1. This was the beginning of my project. In 2A, I'm working on the end of it. And then the 4-fragment for 1A and 1B is still in preparation. So overall, we know that the desiccation assay suggests that channel 2A plays an important role in drought survival, and that Osca 2A may also be involved in salt stress since water agar had too few salts. And the progress towards creating the Delta Osca 1C will allow us to have double and triple mutants in the future, which is very exciting. And the experiments from this research established the utility of moss as a model system for studying mechanosensitive ion channels. So, any questions? Yes.
We're going to have to repeat questions.
I can.
But so I have a really great. Wild type seems to be, ion cells seems to be clustered together.
Mm-hmm.
And then in the mutants, are you observing the effect of proximity? Of these cells, is it possible or pushing ions out could
That's a really good question. So Dr. Genin asked if we looked at the density of cells that survived from desiccation. The desiccation was very new, so we did not really look into density per se, but we did notice that the mutants appeared a little bit more crispy or crunchy. And especially when we were making the salt plates, you have to ball them up into little samples to get them to grow on the different media. And so that was like a slight phenotype, we think, but density is something we should absolutely look into.
Question from actually a couple. Yes. Does the moss contain bonded ion channels that are are these or are there other competing channels?
We know OSCAs are highly redundant, and so that is something we looked into. Is we said, okay, if we remove one of them, like 2A, then enough of the plant's integrity should still be there and it should be able to survive. So that's why in the desiccation assay, there is still some survival because it is still able to recover. But the reason that it's less could be that it's this channel. So we're really studying like each channel significance.
So Chris, I don't know if you can hear me. Can you unmute and ask that question? It'd be great.
Hi, I was just chatting. Can you hear me?
Yes.
Yeah, great. Yeah, I cannot hear when audience members speak. So speakers, presenters, be sure to repeat anything spoken in from an audience member. I vaguely heard a guy tell me to unmute and ask the question. I guess I had two questions about multiple ion channels. Are there other ion channels that are capable of this function that you might have not knocked out?
I'm only aware of OSCAs using calcium, but there could be. I'm not a biology major, just to clarify, in case anyone was wondering.
Yeah, no worries. I guess another question I had was I was curious about the CRISPR-Cas9 targeted system in your materials and methods figure 3. You show that you're targeting within the pore sequence, and it looks like a little bit on either end is left after the gene edit. Why not take out the whole sequence? Why are you targeting within the sequence and leaving some behind?
So with CRISPR, there's always the risk of it cutting a few base pairs in the wrong on either side. And so we wanted to make sure that we wouldn't disrupt any of the other genomic sequence in moss. And we said, if we just remove most of the pore, it'll disrupt the gene's function and we can make sure we're not causing any other mutations on accident.
Excellent. Great answer and great presentation. Very nice.
Ask one more question of the session. The only part of the talk that was confusing was that there were two things going on. There was crispy and CRISPR.
Exactly.
And so can you, those of us who've never seen crispy moss before, a sense of what happens to the moss when it's desiccated? And I'll repeat the question.
So Guy asked what happens when, like, what the moss looks like when it's desiccated. So if you actually look at the picture in figure 6, the water agar, like that's like the most wet I've seen moss. And so it's just, you take that moisture and it's completely gone. That's what we let them dry out all night. The edges will actually curl and like, because we grow moss on cellophane, so that way it doesn't get stuck in the media. And after we let it dry out overnight, even the edges of the cellophane were curling. It was so dry. And then we put it on the BCDA tea media and let it rehydrate, like press down all the edges. And it didn't rehydrate like nearly as much as on the water agar, but the picture in figure 4 is pretty close to what it looked like.
How crispy is it?
I don't know how to quantify crispy.
You're a mechanical engineer.
I know. They don't teach me that.
What does it remind you of?
Brittle?
Liz said it could be friable. It's like friable. Mhm. Dr. Haswell is just telling us about how this is like very new research into mechanosensitive channels in MOS desiccation. So that's exciting. Thank you.
Great. Thank you so much, Julia. Okay, our next presenter is Annika Avula, and I'm going to get her slide up. Okay, come on up, Annika. Annika is from Central College, and she worked this summer in the Dixit labs. Oh, hang on. That's not sharing. There we go. There we go.
Thank you, Patricia. Hello, everyone. My name is Annika Avula, and I'm a rising junior at Center College in Danville, Kentucky. I'm currently a mathematics major, and I was fortunate enough to work in Dr. Ron Dixit's lab this summer with my bench mentor, Ryan Calcutt. So plants are not like us. Plant cells have the same environmental stressors that we do, but they can't move and their cells are glued together. So with pressure from the inside and outside of the cell, plant cells are constantly growing. There's lots of stress, but we have no idea how plant cells feel in response to that stress or what they do with those inputs. So we're interested in studying how plant cells respond to mechanical force. In response to this question, my bench mentor, Ryan, and I have been working on creating a model system in vitro, so outside of the plant system itself, for analyzing how individual plant cells feel, generate, and respond to mechanical forces. In particular, not much is known about how cells respond to these forces on the single molecular level. So this project involves 3 main steps. The first step is to find a flexible substrate that cells can stick to. The second step is to develop a microfluidics device to house this substrate with our specimen. So this will help keep the cells alive for longer term experiments in a controlled system. The third step is to apply force to the cells via the scaffold in the device and observe the effects. So going back to the first step in the project, flexible scaffolds were created that the model plant cell that we use, the BY2 tobacco cell, could stick to as they grow. Previous work in the lab led to the development of a new protocol for co-incubating the tobacco cells with sterile scaffolds to allow for growth and determine which scaffold was the most effective. It was found that the most effective scaffold was the nano-aligned PVDF-TRFE, or NAP scaffold. And this scaffold seemed to exhibit properties that were most similar to the plant cell wall itself. So we know that the cell wall plays a large role in cell-to-cell adhesion, but it hasn't been confirmed yet which part of the cell wall plays the largest role. So to investigate this question, we created one of our secondary goals, which was to generate cell wall mutant calli. A callus is a cluster of undifferentiated cells. from a different model plant called Arabidopsis thaliana. And the advantage of using this plant is that it already has a lot of mutant lines that have been developed. In addition, we wanted to see how the alignment of the fibers on the scaffolds affected the alignment of the BY2 cells as they were grown on the substrate. However, as we began culturing our BY2 cells for this experiment, we found that they only grew to a certain point and then would not grow past that to maturity. So we tried numerous methods of helping the cells to grow. We systematically went through the list of ingredients. We changed the list of ingredients. We changed the pH of the liquid media that the cells were being cultured in. We also changed the initial concentration of cells in the media. But in the end, the only thing that worked was moving the cells to a different location where we could control the temperature and increase it to 25 degrees Celsius. So under method development, you can see the pictures of the cells in flask. The top picture is what cells in stasis look like. They are more translucent, kind of an off-white color, definitely like not as bright as the mature cells in the flask in the image below. And when I did the live dead assay on the cells in this flask, I found that a large percentage of the cells were not alive. So the magenta dots represent dead cells and the light blue circles represent cells that are alive. In the bottom image of the cells in flask, so those are cells that have developed to maturity. They are more dense, definitely less translucent. You can't see through them, a brighter yellow color. And when I did the live dead assay on these cells, I found that they were happier, they were healthier, growing in their characteristic cell file formation. And there's really only one dead cell that you can see present in that image. So while much of my summer was focused on rehabilitating BY2 cells, we did develop an initial device design for our microfluidics device. So our preliminary device design is seen in the third image under experimental goals, and that design features a dual-sided specimen chamber with vacuum chambers on either side. When we developed our new proof of concept with the Meacham lab, we decided to focus on a single-sided specimen chamber and got rid of those vacuum chambers since they weren't really necessary for initial testing. So our model has PDMS as the base. And PDMS is a clear polymer that our scaffold will sit in. It's non-reactive, it's oxygen permeable, which is super important for plant cell growth. And it is molded to the channel. Above that, we have our NAP scaffold with our specimens, so our BY2 cells. Above that, a media channel, and to top it off, a glass cover slip that provides rigidity and will allow us to image the cells through the device. But we want to be able to see what's happening in the cells as we image them. This involves generating DNA constructs that code for fluorescent marker proteins that are indicative of plant responses to mechanical stress. So the first two responses are rapid responses. They're calcium and pH. And in the reference image on the left from the Genin lab, we have a trichome that's being bent. So a mechanical stress is being applied to it. And in image B, you can see that the surrounding cells begin to glow. And this is a result of the fluorescent marker protein that is indicating that pH change as a result of that stress. The last two responses are slower responses. It's microtubule orientation and cell polarity, and those happen over hours to days. So to conclude the summer, we found that BY2 cells are happiest and healthiest when they're cultured in 25 degrees Celsius in a temperature-controlled environment. We designed a preliminary chip culture device, and we have a few ongoing investigations. So our Arabidopsis thaliana cell wall mutant calli are currently being tested for scaffold adhesion. The transformation of our BY2 cells using the DNA constructs that we generated is upcoming. And we had a side project going where we were looking at phenotypes for mutants in microtubule bundling proteins. And we saw that one of the mutants in the MAP65-7 gene seemed to affect Arabidopsis roots, skewing them to the left. So that image that you see there is mirrored, but we only did this experiment once, so further replication is necessary. Future directions include further characterizing cell adhesion to the scaffolds, using BY2 marker lines to further examine cell physiology, and finally, building the first plant tissue on a chip with the Meacham Lab at WashU, a feat that is long overdue for the field of plant mechanobiology. And with that, I would like to thank the NSF for funding this REU, the Dixit Lab, and my bench mentor, Ryan Calcutt. And I will take questions now.
This microphone may or may not work. Anybody? Is this working? All right. So I have to repeat the questions. So Liz Haswell from Washington University in St. Louis.
Lines we use, and if the process of making cali from those mutants was we found to be a little different. So the mutant lines that we looked at were QA1-1 and XXT1 and XX. We didn't find that the process for generating cali was any different. I did find that kind of the quality of the cali was a little different. Like the mutant cali were definitely harder. It was harder to find like, so cali are supposed to be like a little softer. They're supposed to be kind of like more of an off-whitish kind of color. And the mutant cali started to turn brown a lot faster. Yeah, so they were like crunchy and crispy. They were a little harder. So definitely it was like harder to find cali that we could like harvest. Thank you.
I have a question from a mechanical engineering perspective. I'm going to speak loudly enough that I hope people can hear me through the microphone. Apologies if I injure your ears. So when mechanical engineers see something change as a function of temperature, one of the first places mechanical engineers look is at viscoelasticity. And so viscoelasticity of the cell wall is also affected by pH. So the question is, does the optimum temperature for growth change with pH in the solution?
So Dr. Genin asked if the pH changed with the temperature.
So is the optimum temperature for growth? When you find this 25 degrees Celsius, does that, if you change the pH, does that optimum, if you change the pH, which can change the stiffness of the cell walls,
right?
Then does that optimum temperature change?
I see. That is a really good question. I think that would be a great thing to investigate. Yeah, so the question was basically if you change the pH of the cell wall, does that change the temperature that the cells can grow at? And he was making a comparison between like the BY2 cells growing at 25 degrees Celsius and if we change the pH, what that would affect. And I honestly don't know the answer to that question, but thank you for a new question to investigate. It is fun.
Any questions from online? Can you hear the audience questions now? Is this getting transmitted through Zoom?
Yeah.
Yes.
Yeah, excellent. And I have one more question, but I just want to check if there's any online. And so I would like to ask one more question, if the session chair will allow it, which is that in the device you've developed, you have this specially designed material on one side, but then PDMS on the other. Do the cells interact with the PDMS at all, or do the cells closer to the PDVF? And I couldn't actually read the rest of it even on my computer. The special scaffold that you have. Is there a difference in the cell survival and cell or any other aspect of the cell depending on where they are in that channel?
I don't think that there's too much interaction with the PDMS besides like just making sure that oxygen can get to it and stuff. It's more just like something that the cells will sit in on top of the scaffold. So it holds the scaffolds which the cells sit on. And so, yeah, I mean, I don't think there's too much like real interaction going on there.
Great. Well, thank you very much.
Thank you so much, Annika. Okay. Our next speaker is Abdulaziz Said, who worked in the Amit Pathak lab this summer. Shared yet, Abdulaziz? Okay. Come on.
Hello, everyone. My name is Abdulaziz Said. I'm a rising junior at Washington University studying biomedical engineering. This summer, I've been working in the lab of Dr. Patak under the mentorship of Jose Almeida and in collaboration with Cameron Bleem. Today, I would like to discuss our projects and our relevant findings. First, I would like to discuss some important background information important to understand our project. Then I would like to discuss our specific experimental design and results. And lastly, I would like to finish with a quick discussion of the future outlook of this line of work. A great deal of research has been devoted to trying to understand the underlying mechanisms in breast cancer disease. A large contributor for mortality in many patients is the spreading of a tumor throughout the body to cause metastatic disease. In past studies on the topic, signaling pathways involving REU kinase and the REU-A-G protein family have been shown to help facilitate cytoskeleton remodeling in tumor cells in such a way to cause these cells to become more malleable and better able to undergo cellular migration. In subsequent studies, there were found correlations between low oxygen content in a tumor's microenvironment and the subsequent development of metastatic disease due to a possible interaction with the REU kinase signaling pathway. So this leads me to our study this summer. We set out to try to understand some specific effects of hypoxia, which is low oxygen content, on cancer cell behavior. And we hypothesized that the hypoxia will allow the cells to be, cancer cells to become more active and better able to undergo migration. In our experiments, we focused on two major cell types. The first are MCF10As, which are healthy breast epithelial cells. And we use BT549s, which are triple negative breast cancer epithelial cells. After culturing the cells, we then developed a set of collagen gels with 200 microliter volume and approximately 2 milligram per milliliter density. After the development of the collagen gels, we seeded each gel with approximately 25,000 cells from each of the two types. Once seeding was complete, the samples then underwent priming for 24 hours in either a normal oxygen environment or a hypoxic oxygen environment. This led us to having 4 distinct experimental groups, where groups 1 and 2 were primed for 24 hours in normoxia, which is approximately 21% oxygen, with group 1 being the healthy MCF cells and group 2 being the cancerous cells. In groups 3 and 4, we primed in hypoxia, which is 5% oxygen content for 24 hours, with group 3 being healthy and group 4 are the cancer cells. After priming was complete, we then conducted a time-lapse study for approximately 4 to 8 hours by imaging the samples with a Zeiss confocal microscope. The time-lapse study allowed us to gather some data on understanding how the cells behavior, how they move over time, and their migration behavior. Once the time-lapse study was complete, we then fixed the cells with formaldehyde and took some bright field images for a later phenotypic analysis. That concludes the methods for our experiment. We can now move on to our results. We can start with figure number 3 on the poster. This figure is showing 2 bar charts. The bar chart on the left, labeled mean cell area, is comparing the cell area in our 4 experimental groups. The right bar chart is shape index, which is a measure that we compared with the 4 groups again. We found a pretty clear pattern with the healthy MCF cells showing a decrease in cell area with normoxic going to hypoxic, with the area decreasing from 1600 micrometers squared to about 690 micrometers squared. The shape index for these cells also decreased from about 6.6 to 3.9. For the hypoxic cell, cancerous cells in hypoxia, these ones showed a very dramatic increase in cell area, almost or more than 3-fold increase in area, going from 970 micrometers squared to 3,700 micrometers squared, with the shape index also going up from 3.8 to 6.4. These findings are important as they show that with the increase in area that the cancer cells are having, that the hypoxia seems to be allowing them to become more engaged with the collagen environment and become a little bit more viable. Moving on to figure number 4 on the poster, this is our migration analysis that we conducted after the time-lapse study. These figures are heat maps that are showing the collagen deformation of our gels over the time-lapse time. We found that the maximum collagen deformation speed was about 250 micrometers per hour, with the cancer cells primed in hypoxia showing a very dramatic increase in their velocity, while the healthy cells showed a very small increase in velocity going from normal oxygen to the hypoxic oxygen. Our last figure is figure number 5. This is of our phenotypic analysis that we conducted with the 4 distinct groups. In this figure, the cells are highlighted in yellow. We saw in this analysis that the MCF healthy cells seem to become more round in the hypoxic environment compared to them in the normoxic environment, while the cancerous cells in hypoxia look to have larger protrusions. This fits with the earlier finding with cell area, in that the cancer cells with their larger protrusions seem to be more engaged with the collagen gel environment as opposed to priming them in a normoxic condition. This concludes our results, where in total we found some specific effects of hypoxia on cancer cells that hypoxia does seem to have a positive effect for cancer cells, helping them to possibly become better able to undergo migration. In the future, these studies should look more into some of the specific pathways and signaling pathways that are getting affected by hypoxia to lead to these phenotypes. Further studies should also be conducted on the behavior of groups of cells. While in our studies, we looked at single cell behavior, it is important to understand breast cancer pathology to look at behavior of collective groups over their migration behavior. With that, that's my end of my talk. Thank you to everyone for listening, and thank you in particular to Dr. Patak for giving me the opportunity to conduct this research this summer.
Thank you, Abdulaziz. Any questions from the audience, or can I start with one? I'll start with one. So your plot, the heat map that you developed with the speeds of contraction, so that there's much greater speeds, so presumably greater contractility in the bead distribution underneath the cancer cells, if I understood it correctly.
Yeah.
So what about the magnitude of how much the cancer cells pull on the substrate? So they're pulling faster, but are they also pulling more in hypoxic conditions than do the healthy cells?
Yeah, that's an important detail. We didn't look specifically at, we were trying to look at what changes in hypoxia as opposed to normoxia, and if the cancerous BT cells, if they have better better motility. So we were looking more on the changes and not the specific magnitudes, although that is important for further investigation.
Well, my next question is there such a word as hyperoxia? And if there is, what does it do?
I'm sure there is a condition with excess oxygen that I'm not sure what that would cause. I would presume more detrimental than toxic environment.
It'd be great. So if the cure to all cancers was just having a lot of oxygen, that'd be really convenient.
That would be convenient, yes.
Can I ask one more question? So let me double check to make sure I'm not hogging the microphone, which I am, but hogging it from those online, which I'm not. So the images that you have in Figure 5, these are very striking images, which show that for healthy cells under normoxia and for cancer cells under hypoxia, they're very elongated, whereas the cancer cells under normoxia and the healthy cells under hypoxia are very rounded. But neighboring each of these elongated cells, there's in the pictures that were chosen, there are a couple of cells that actually stayed kind of rounded. And so the question is, if you look at the cells that normally stay rounded, the hypoxic healthy cells, the normoxic cancer cells, are there a fraction of them that also end up becoming highly elongated? Is there a lot of variability from cell to cell?
There was a little variability. We would, to solidify the results, we should conduct more trials. We did see the cancer cells in a normal environment. They were very largely, almost completely very round and they don't seem to really be engaging with the collagen at all. The healthy cells were pretty well at elongating and creating protrusions to connect to the collagen in the normal for most of them. So we did see a pretty clear trend.
Well, thanks very much. And a round of applause for both Cameron and Abdulaziz. Well done, both of you guys.
Hi, Guy. Are we still open for questions?
For you, we are open all day and into the weekend. Professor,
I just want to, yeah, I'm so sorry I couldn't be there in person. I just want to comment how amazing Abdul and Cameron both did. Just to note that this is an entirely new project. So in 2 months, they were able to establish hypoxia and then get all these results, which is, I think, remarkable. Sometimes, we try a new thing, a new project, things don't work out for months and months, sometimes years. So I just wanted to make a note of, how fantastically well this undergraduate summer research went with Abdul. Yeah.
Thank you.
well done to both of you guys, and thank you for those comments, Amit. Great. So Abdulaziz and Cameron, congratulations to you both.
Thank you.
All right, thank you so much, Abdulaziz. This is the time when we have a break on our schedule. Do we want to go ahead and take that break? Yeah. Okay. Right, so those of you who are present in this room, there are some snacks. I think there are rules about the snacks, so please observe them, whatever they happen to be. I have lost track of what the snack rules are, but
so snacks are out loud in here. For those who are at Bayer, please get up and walk around a bit for the benefit of the mechanobiology of your body. We'll be back in a few minutes. For those who are here, the rules are the university does not allow you to pop the corks of champagne bottles when they're pointing towards people. And please be careful with the caviar, because the caviar is very salty. You can find yourself dehydrated. We'll be back in a few minutes.
Okay. Right. Good morning, everybody. How was the break? I was in 2 labs this summer, Dixit and Genin lab, and this was what I did. The organ level root skewing is highly sensitive to increased shear force in the right-handed twisted mutant spiral 1 tag 3. So we already heard that plants have mechanisms that allow them to adapt to environmental forces. This called tropisms. Can go towards or away from stimulus, this being, for example, touch or pressure or light. These mechanisms can be seen more obviously in the shoot system, but they're also present in the root system. So plants direct their root growth towards nutrients and water and avoid obstacles to continue their growth. And direction of root growth is determined by the orientation of the microtubules or the microtubule cytoskeleton. In normal plant cells, microtubules arrays are perpendicular to the cell on the cell wall. And this array allows the root to grow in a vertical manner, and thus looking like it was stretching on its y-axis. It can be pictured as if you were holding any toy and you squeeze it like this, it will only have a way to grow vertically. In the Dixit lab, we work with the model plant Arabidopsis thaliana, more specifically the Columbiana/0 genotype, or Col0, to study the mutations that affect microtubules, mutations that often lead to skewed cell growth. A very noticeable mutation in the twisting and skewing of the roots. There are many twisted mutant genotypes, but the one we used in the lab was a spiral 1 tag 3, which lacks microtubule plus and binding proteins. Now, this mutant not only shows skewedness of the roots, but also skewedness of the cell files. So if you look at figure 1, A and C are Col0, and B and D are spiral 1 tag 3. You can see in the agars same concentration. So Col0 grows straight and the cell files look straight, but spiral 3 has the right-handed skewedness and also do their cell files. Previous work done by my bench mentor showed that increased shear force affects organ level root skewing in twisted mutants. And the natural tendency of spiral 1 tag 3 mutants roots to lean to the right seem to be affected by the different agar concentrations. Organ level skewing was obvious, but cell level skewedness has not been fully studied. So we wanted to see if the orientation of the mutant cell files would stay the same through all different agar concentrations. And that was our hypothesis. as if the change in cell file skewing will match the root skewing in all different concentrations. So for this, we repeated the experiment. The experiment was to sterilize col0 and spiral 1-tag3 seeds for 48 hours. Then the seeds were sown in planes containing 3 different agar concentrations, 1%, 3%, and 5%, and they were grown in long day conditions for 3 days. Then the root cell files of the seedlings were imaged using like a bright field dissecting microscope and analyzed using image J, and finally using a graph pad prism to obtain the results. And the results were that in image figure 2, you can see on the left side, you can see col0, and on the right spiral 3, that is the root skewing or organ level skewing. So for col0, they were all straight, no skewedness, and for spiral 3, you can see the changes in agar concentrations. 1% represented in black shows the right-handed skewedness. 3% agar shows an almost corrected vertical growth, and in 5% it shows an unusual left-hand growth. And in figure 3, you can see the cell file level, which indicates for col0 and spiral 3 in all the same percentages of agar. But you can see that for col0, the cell files, they were all also straight. And for the different percentages in spiral 1-tag3, they were all also the same, no statistical difference. So they were all still skewing to with the left-handed orientation. I also work in the Genin's lab, trying to replicate it with a model. There you can see figure 4, the twisting from the molecular level, what the forces would be, or the forces implemented in the root that are needed to correctly replicate the model. On the left side, you see the wild type, how microtubules in green can squeeze a cell and make the cell roll vertically. And when microtubules are skewed, the cell file is obligated to skew also. So figure 5, the forces present in the bending of the root, you can also see that while this, because the root is 3D, so you'll have compression and tension on one side, and when these two meet, because it's also continuous, it will create the twisting motion that the roots are showing. And so for a conclusion, we concluded that the forces surrounding the root will have effects on the root development, and that cell skewing, it's uncoupled from root skewing. The analysis of the microtubules, it's necessary also. We had this in mind planned, but due to time, we did not have enough time to complete the experiment. And for the model, we also need anisotropic material properties, because the model right now is isotropic, which is not a good representation for the root. Thank you. I'm open to questions.
Thank you, Osmar. So I would actually like to ask a question, which is about how about the experimental side, about which I know very little, but I find this a fascinating system. Do you have any thoughts on where what physicists call symmetry breaking occurs? So for living systems, it's very unusual. Something special has to happen for symmetry to be broken. And so somehow there's some aspect of these cells that causes the microtubules, the cellulose to be laid down in the spiral pattern. What are your do you have any thoughts on what protein structures underlie that breaking of symmetry, so it actually develops this chirality?
The way I think, do I have to repeat the question? All right.
So what proteins? How does the so you've shown how the twist leads to how the lack of anisotropy leads to excuse me, how the anisotropy leads to this twisting of the root. But what leads to this anisotropy? What causes? What are your thoughts about what causes the twist to occur in the first place? The twist that you see in Figure 1. Do you have any ideas on that? And I'm pretty sure the answer is that nobody really knows this yet fully. But is there anything you came across in your reading this summer that you find find to be a potential source of all this, or is it still kind of an unknown?
I think it's just the way I understood it is the microtubules guide the cell files. So the cell files will create this chirality because they occupy space. So just by occupying space, if one is skewed, the rest of one will also need to be skewed.
Liz Haswell is about to either make up stuff or educate us, or both.
Yeah, I don't really know, but I had the impression that there actually was some intrinsic anisotropy to tubulin itself. And so that as microtubules were formed, there's a certain spin to them, and that people had sort of interpreted that might be amplified at the level of the cell and then subsequently at the cell file. I think something else to think about is that you're imposing anisotropy by the fact that you're growing these on the surface of an agar plate, right? So that's not really how they evolved to grow. I mean, it's possible, but my feeling is that Arabidopsis didn't evolve to grow on the surface of an agar plate, but that we also see them doing this like circular movement in the soil. So there's a recent publication where people have watched roots grow in the soil, and they also do this sort of mutation thing. So it's very interesting to think about what you might be replicating and what you might be missing when you grow roots on a solid support. Not really a question.
Observation, yeah.
But it's raising important questions in the field. Thank you very much. All right, and the last piece is, could you explain how is it that so what goes on? How is it that you end up with a twist to the side with the same stuff on the same stuff on the top and on the bottom, just kind of rotated around? How does stretching that lead to the twist in one dominant direction instead of opposite directions?
Because the cells will be kind of rounded. So when, for example, in Figure 5, when one side is being affected by compression, you can think about it as a box of straws. If you stretch the box of straws, it's gonna create these straws. They're gonna be vertical. So if you apply pressure to those both sides, the box is gonna create it's gonna flatten out these straws. So one side is being compressed, the other one is in tension, and that creates that spinning.
Beautiful. Well, thank you very much.
Okay.
Thank you so much, Osmar. Okay, our next presenter is Miranda Copenhaver from Washington University in St. Louis. I'm going to have 2 claps. Okay, go ahead.
Hi, everyone. I'm Miranda Copenhaver. I'm a rising junior here at WashU, and I'm majoring in biomedical engineering with a minor in computer science. And this summer, I had the pleasure of working in Dr. Marcus Foston's lab with my bench mentor, Morgan Lee. And really, the heart of what I was doing this summer is material science. So I was making what I call cellulose nanocomposites, and I'll explain a little bit more about that in a minute. And I was making them with a chemical called benzophenone through UV activation. So the material at the center of my research this summer is called cellulose nanocrystals. I call them CNCs for short, and they are exactly what they sound like. When cellulose forms, it's a polymer chain of glucose, and that chain can be amorphous and it's all jumbled up, or it can be in a really nice organized structure called the crystalline region. And cellulose nanocrystals are just the crystalline portions of that cellulose. And we've been taking those CNCs and combining them with proteins to create what we call nanocomposites. And these nanocomposites are materials that have a really wide range of applications. First and foremost, the application I'm most interested in is that they are biodegradable food packaging. If you were to look in the average American household and look in their garbage can, the number one thing you would see is single-use non-biodegradable food packaging. So this offers a really interesting alternative. Additionally, nanocomposites can also be applied to the medical field for tissue repair and drug delivery, and they can be applied to industry for adhesives and anti-reflective films. Aside from the really wide range of applications, they also have really favorable chemical and mechanical properties. So CNCs are really strong. They have an elastic modulus close to that of steel. So when we reinforce proteins, typically in my lab soy proteins for the food packaging application, when we reinforce proteins, it's like reinforcing concrete with steel rods when you're building a structure. And they also have a really high surface area to volume ratio, which means that there's just a lot of surface that you can use for functionality. So my research this summer focused on using a novel technique to create these nanocomposites and create the crosslinks between CNCs and soy proteins. And that process used benzophenone, which is a chemical you might not know that you know it, but you do. It's used in a lot of sunscreens because it's UV activated. And the reason that this process is really interesting to us is because the crosslinks that are formed from this process are covalent bonds, whereas other processes that have been used in the past have been more so like intramolecular interactions that aren't as strong. And you can also use the UV activation in a more precise way to control where in the substance you are causing these crosslinks. And so the goal of my research was to confirm that this process actually works and to look into the effects on the mechanical properties of the films. And that figure 1 right below is just a picture of CNCs to give you an idea of what they look like. They're long whisker-like shaped structures. So as far as methods go, the process of making these nanocomposite films is really a process of building outwards. You start with your CNCs, which you extract through acid hydrolysis. And then on top of that, you add your tempo, which is just a chemical that oxidizes the surface of the CNCs with carboxylic acid, and that just adds more functionality. And then you add your benzophenone, which I'll refer to as BP, and that's our magical UV activated chemical. And then from there, you add your soy protein, and you pour that into your nanocomposite films and then expose it to UV to cause the cross-linking. And figure 2 in the middle there is just a picture of each of the reactions at each step of the process. So your oxidation with tempo, BP coating, and then exposing it to the UV excitation to cause the covalent bonding with the soy protein. So the first portion of my summer was spent more so focusing on the tempo. It's really important to note that in this process, each step is limiting to the next because you're kind of building outwards. So if you don't add a lot of tempo, you can't add a lot of BP. And it was important to make sure that we were optimizing the amount of tempo we were adding. So if you look at figure 3 at the top there, that's just an example of one of the many conductometric titrations that we performed. We had a lot of graphs that looked exactly like this. And what that tells us is the linear decreasing region is the sodium hydroxide that we added to the solution using up various ions floating around in our solution. And then the middle flat portion is the sodium hydroxide actually reacting with the carboxylic acids on the surface of CNCs. And then the last linearly increasing region is the sodium hydroxide just adding extra ions to the solution. And the most important part of this is the middle flat region, because that's the part that tells us how much carboxylic acid is actually on the surface of our CNCs. And so we took many graphs like this, and then we extrapolated the data to get a really precise idea of like what that middle region looked like. And Figure 4 on the bottom there is a summary of all the titrations we did and at different tempo loadings. And what we found is that after a certain point of tempo, you just kind of get diminishing returns and it plateaus off in terms of carboxylic acid coverage. And we found that the optimal point was about 1.6 millimoles of tempo per gram of CNC. And that was the first portion of my summer, was optimizing tempo. The second portion of my summer was used up mainly confirming that this benzophenone process we started actually worked. We wanted to make sure that the covalent bonds were forming where we wanted them to. And we tested that through water absorption. So we exposed the films to 95% relative humidity for 2 days. And if you look at figure 5 at the top, the 2 bars on the right were films that had no benzophenone in them. And when exposed to the 95% relative humidity, they over doubled in weight. Because they absorb so much water, and that's because they don't have those covalent bonds. And when you have your covalent bonds, it packs the components in a lot tighter and it makes them less able to absorb water. And when you add in your benzophenone, you see a little bit less water absorption, but that's purely because benzophenone is less hydrophilic than your carboxylic acid. And when you get the least water absorption, that occurs when you have benzophenone and UV activation. And that's really key, is that you need both components to cause those covalent bonds to really decrease your water absorption. So that result in figure 5 confirmed for us that the covalent bonds that we wanted to see were indeed forming. And from there, what we want to do is kind of play around with the recipe that we've created, change the UV exposure in terms of intensity, duration, and using photomasks to expose the films to UV in different parts. And then also play around with BP loading, and then further use imaging techniques to look at the cross-linking at a molecular level to really get a good idea of what's happening. I'd also like to thank the Center for Engineering Mechanobiology and my cohort for all of the support they've provided, and the Foston Lab and my PhD mentor, Morgan Lee, for just teaching me so much this summer. Thank you.
Can I start with a question?
Go for it.
One of the things I find fascinating about the mechanobiology plant walls is that the plant wall can change in response to its mechanical history. And so the question I'd like to ask is, does that matter in terms of turning the plant wall materials into something that's useful in the engineering sense? So are all of these, does cellulose nanocrystals change at all depending on the source? Would a plant that has a really thick and stiff stalk have different cellulose nanocrystals once you break it down that much than the blade of grass?
Yeah, so the source of your cellulose actually makes a really big difference in what your CNCs look like. So you can get cellulose from a lot of different sources. It's everywhere. And you know, algae will produce a different look than cellulose nanocrystals from plants, and they vary in length mainly. So these ones you can see are around like 300 nanometers long, but they can be super, super short, like 30 nanometers long, you know? And also the extraction process that you use to get the CNCs in our lab, and typically you use sulfuric acid hydrolysis. You can also use hydrochloric acid. The reason that we use sulfuric acid is because it leaves the CNCs with a higher surface charge, which makes them more easy to disperse in water, which is important because you want an even dispersion of your product when you make the film, because you want consistent testing.
Thank you. So do this, you raise your hand. Do you have a question? Oh no, sorry, I missed up. In that case, can I ask one more?
Yeah, go for it.
So did you have a chance to look at the effects on mechanics? So certainly water absorption is one great way to tell whether or not you have really strong and firm bonds between molecules. But then the stiffness is also a way to do that. Have you? What are some? I guess there aren't data up here for stiffness. What are some of the challenges for actually doing mechanical measurements on these systems?
Yeah, so we actually did try to do some mechanical testing this summer. It gave us quite a bit of trouble. The films they're they're films. So they're really thin and hard to pull out of the Petri dishes. And we were also working on getting a dog bone shape for our films to test it, because that's the ideal shape for your mechanical testing. And we just ran into a lot of problems with actually getting them to dry in that shape. We had like dog bone shaped molds, and when you poured the solution in and let it dry, it would evaporate up the sides and it would give you more of a dog bone cup than a film. But in future experiments, we definitely want to look into the mechanical properties. That is kind of our main interest. This was more of just a proof of concept to make sure the covalent bonds were forming.
Fantastic. Thank you very much.
Thank you.
Okay, thank you so much, Miranda. And our next presentation is Sorina Munteanu from the University of California, Merced.
Hello, everyone. My name is Sorina Munteanu, and I am from the University of California, Merced, and I'm a rising senior studying bioengineering. So this summer, I had the opportunity to work in Dr. Genin's lab under my mentor, Shangjin Pang. And a little bit of background information on my project is that mechanical stresses on and within the actin cytoskeleton are critical determinants of cellular function and morphology at the earliest stages of cell and extracellular matrix interactions. And these stresses will resist microtubule growth, which in turn generate protrusions that are required for extracellular matrix remodeling. And in turn, with being able to manipulate the cell ECM interaction and remodeling, we will have an enhancement of answers for fibrosis and wound healing. And in a previous study in the lab, the researchers investigated 3 out of 4 of components within the cytoskeleton, which are actin, myosin, and microtubules. And in figure 1, what this figure shows is that as the actin transmits mechanical forces, it will attach to myosin, which is a motor protein, and cause tension. And then microtubules will work under compression. And so microtubules and actin work as a function of one another. And what we have seen is that before there's actomyosin contractility, there will be less restricted microtubule growth, and as a result, no contractile force. But then when there is contractility, it will lead to microtubule growth. And in our investigation, as this was done under a 3D microenvironment, so with collagen, we looked at the 2D microenvironment, so a media substrate. And we are hoping to see if that we'll see similar results as was done in this previous study. So a part of the experimentation was recovering the cells and passaging them in order to transfect them with a tubulin GFP. And with that tubulin GFP, we're hoping to observe the microtubule growth and its relation to actomyosin contractility. And in my results here, you can see that we are able to get the fluorescence of the microtubule growth and under different drug treatments. So for the untreated cells, as actomyosin contractility is still involved here and works in concert with microtubules, there will be protrusion growth, but actomyosin contractility will cause a resistance towards that growth from continuing to grow. And then with the cortisol, we inhibit the microtubules. And as you can see that there is no protrusion growth. And for Y27632, that inhibits REU kinase, so it affects cell motility and shape,
And we're able to see that there are longer protrusion growths, more branches, and cell polarity. And then with blebbistatin, that inhibits myosin 2, so inhibiting actomyosin contractility as well, and it shows similar results to Y27632. So longer protrusions, polarity, and more branching. And so I did a computational analysis, getting quantitative results for these treatments right here, and using ImageJ and AIM browser to visually as well as quantify the length of the protrusions in micrometers. And so this graph on the left for figure 4 shows the average number of protrusions. And what we can see here is that there is significant difference between each drug treatment. So 1 asterisk indicates a p-value of less than 0.05, while 2 asterisks indicate a p-value of less than 0.01. And so what we can see is that there's a high significant difference between the control and nicotazole, the control Y27632, and nicotazole and blebbistatin. And so for the average number of branches, there is no significant difference between Y27632 and blebbistatin. And then for the average protrusion length, we can see that Y27632 and blebbistatin have a high significant difference for the average length, as well as nicotazole and blebbistatin and the control and Y27632. So to conclude, we are able to observe and see that in a 2-dimensional microenvironment, there is a competition between actomyosin contractility and microtubule growth. And we now have a clear understanding as well as that actomyosin contractility cannot work independently and must coexist with microtubule growth in order to create this balance of tensegrity. And so from these observations, we can potentially manipulate the extracellular matrix to promote wound healing and potential therapies for fibrosis. And this last figure here is just a final schematic to show or represent what is seen in figure 3. So for the control, since there is a contractile force, there will be restricted microtubule growth as there's still growing protrusion tip, but then the actomyosin contractility will lead to the microtubule protrusions having to depolymerize and will cause spreading. And so for nicotazole, there will not be protrusion growth. And for the latter 2 drugs, we are able to see that cell polarity and longer growths. And so for future studies, what we're hoping to do is to test this hypothesis within a 3D microenvironment with different densities of microbeads in order to see how different collagen densities will affect this growth. And we would like to observe this relationship between actomyosin contractility and microtubule growth within suspended cells. So far, we have been troubleshooting with our 3T3 NIH cells that we use, which are mouse embryonic fibroblasts, but these tend to spread on the bottom of the dish. So maybe we're hoping that a different fibroblast will work better in this case. And lastly, we determined the baseline parameters for this project and also supporting the previous results. And we're hoping to order to model a cell behavior for the cell and ECM interaction. So I would like to thank the Center for Engineering Mechanobiology, NSF, the National Science Foundation, the Genin Lab, and my cohort for this knowledge and support and the funding to do my project. Thank you for your time.
Thank you, Sorina. Questions for Sorina? From Shanjun Peng in the back.
Hi. So my question is that can you explain more about the model you use for the first figure? So why the microtubule should be parallel to the myosin, but in series with actin?
Okay, so why they're in parallel is because as microtubules are under compression and contractility is under tension, they are a function of one of each other, and so in their relationship, if you're able to cause compression with the microtubules, it should pretty much lead to contractility for the other, so they're almost opposing each other or polarizing each other. So it wouldn't work in series for that reason.
Thank you. And if I could ask one as well. So you've really shown something new and interesting about a really well-studied, densely studied problem. And you showed that there's this trade-off between actin contractility and sort of outward pushing of microtubules in the early stages of how a cell adapts its environment to its liking. Do you have any thoughts on how this might be applied? So you mentioned this is useful to understanding, say, development in cancer and fibrosis. Any thoughts on ways that you might apply this?
So for previously, it was falsified that we can observe this behavior or this competition between actomyosin contractility and this microtubule-based protrusion growth in a two-dimensional microenvironment. And so with this further understanding and being able to apply this also in collagen or tissue, it gives us a deeper understanding of how we can work towards being able to modulate this growth ourselves and with these access to these drugs in order to potential areas of wound healing. Any cytoskeleton related or ECM related.
Thank you much. Well, with that, thanks, Sorina, and congratulate all of the participants of the 2021 CEMB REU program. So in a moment, we'll ask Phil Taylor from Bayer to say a few closing remarks. But before he does, and also I would like any of the mentors from Bayer who are online who would like to share some sort of parting thoughts about what important next steps are in developing a scientific career. Anybody who cares to is invited to. I would like to say a few remarks myself, which is that this has been a remarkable group of students. This is an incredible set of research for just a couple of months of effort here, especially given that this was the first cohort of students to come through in a couple of years. So this was not a well-oiled machine, but you guys have produced outstanding research. We hope that this has been as meaningful for you guys as it has been for the mentors, and that you continue to be in touch, you continue to develop in your scientific careers, and be in touch as you do so. And so do need some words of thanks. Thank you to Gwen, who's sitting in the background for aside from bringing in caviar and champagne, those pastries that she flew in fresh from Paris on the Concorde this morning were exquisite, and the type of service that we've received all summer long of that sort. That type of effort is deeply appreciated. And so I'd like to give, actually, if you'll give me 1 second here so I could try to share the screen and get this, and instead have your picture up on the main screen, which I'll let our expert Patricia Widder deal with 1 second. So I would like to say a special thank you to all of the mentors at Bayer. This is a remarkable program conceived of by Phil Taylor that's really enabled. It's rare that a student at the undergraduate level has real access to world experts in their areas, PhDs in a world-class business, and it's been truly an honor to have this opportunity to interact. And so I'd like to, for some closing remarks, I'd like to introduce Phil Taylor, who's over there. I'm actually not turning my back to you, Phil. I'm actually turning towards you.
Yeah, thank you, Guy. Yeah, just on behalf of the Bayer team, our thanks to you for the opportunity. I mean, I know hearing from the mentors in the program, they've greatly enjoyed their conversations with the mentees, both on the sort of development and then the science front, and it's been fabulous to participate this morning and get to hear the great talks from the students. So amazing stuff done in a short space of time under some still rather interesting circumstances. I'm sorry that we're not able to get down there and join you in person this morning, but it's been really fun to be part of the day today. And you're throwing thanks at me, which I appreciate, but I did the easy work, right? The hard work was done by the mentors, and so my thanks to all the Bayer team who've been involved, and to the WashU team for working with us and having a fun summer together. Next year, hopefully, we'll really get to do it in person. I know we've said that a couple times now, but next third time's a charm. But thank you.
Fantastic. Phil and all mentors from Bayer, thank you so much for a wonderful summer and for your continued interactions. We'll do our best to save some caviar and champagne for you for next summer.
Appreciate that.
But look forward to being in touch. Congratulations again once more to all of the CEMB Summer 2021 Fellows. So we're going to figure out how to turn this off. Great.
Okay. Thanks everyone for attending.
The recording's off, and now with the recording off, let me again say thanks so much to you guys at Bayer. Thanks for being here, and thanks for being here in more ways beyond physically throughout the entire summer. It really been an honor working with you guys.
Thank you so much. We hope to continue the relationship in many future summers.